Hi, everyone. Welcome to this week's Single Cell and Exploration Club, where we try to introduce some of the features on our platform through the form of a case study. So my name is Julie. I'm a product specialist here on the Turing team. And for this week, I wanted to introduce some of the more advanced features on our platform, BBrowserX Pro. So here we're going to go over a single cell and spatial transcriptomics approach to colorectal cancer research. So I'm going to introduce some features on both BBrowserX Pro and SpatialX. For the agenda today, we're going to look at the colorectal cancer dataset, how you can access it on Talk to Data. And then the advanced features that I'm going to be going over are the cancer cell prediction tool, the cell-cell communication, and the pseudotime analysis. So to introduce the features first before we dive into the demo here, basically what this cancer cell prediction tool does is it's really useful for cancer biologists, people who are studying tumor tissue, because within tumor tissue, there's going to be a lot of different cell types, some that are malignant and some that are non-malignant. So it's really important to be able to tease out that malignant population of cells in order to study them even further. So essentially what this cancer cell prediction tool is doing is it's trying to infer or predict a CNV score for each of these single cells. And it's inferring the score based off of the gene expression information. So for those of you who are not familiar, CNV stands for copy number variation. And this is typically a pattern you see in cancerous cells where they will have high copy number variation, which is essentially where you see proximal genes kind of upregulated in a pattern together or downregulated in some pattern together. And so since we only have gene expression data or transcriptomic data from our single cell data, then we can only infer or predict what the CNV score would be for these individual cells. So the image that you're looking here on the left-hand side, these dark blue cells are high in their CNV score, meaning these are predicted to be the likely cancerous cells. And one of the ways in which our team has approached to validate these prediction results is by leveraging the use of spatial data. So for those of you who are familiar with spatial, this spatial data set is a Visium HD data set with a bin size of 8 microns, which means each of the individual spots can be mapped to singular cells. And so taking that data set of single cells, we were able to run the cancer cell prediction tool, predict which cells were likely to be cancerous. And then when we mapped this single cell back to the Visium HD spots, you can see that the predicted cancer cells are the ones in dark blue here, and they lie at the periphery of the tissue here. Okay. And so in addition to this, this particular tissue was also able to be annotated by an expert histopathologist or a team of pathologists. So this team was able to go in and look deeply into the image, specifically the H&E image. And just by recognizing the morphology of the cells, they were also able to identify which cells were the cancerous epithelial cells. And so this is the image that you see on the right-hand side, where the team of pathologists were able to identify that the cells on the periphery of the tissue were the cancerous epithelial cells. And so now when we compare the image annotated by the pathologists and the image that was predicted using the cancer cell prediction tool, you can see how the predicted cancer cells matched quite well with the spatially annotated cancerous cells. And so this was one of the ways in which our team was able to kind of verify or validate or benchmark this cancer cell prediction tool. Okay. Another thing that I want to mention here is that you'll notice that I have the infer CNV package written here. So the cancer cell prediction tool is based off of the original infer CNV package, but our team has made a lot of enhancements and improvements in order for this package to run a lot faster and also improve the scalability as well. So now the cancer cell prediction tool is able to run on very large data sets. And one of the prerequisites to using this package is that users would have to manually kind of select a reference point for the analysis to run. But one of the enhancements that the team made was that we now allow users to automatically find where that reference point would be. Okay, and then lastly, one of the ways to reduce dropout rates is by grouping cells into pseudocells or metacells. So in the original package, it would be trying to call the CNV score for each individual cells. But we found that the kind of the biological sensibility of the results wasn't that great. And so one of the ways to improve this was by grouping cells into clusters or clustering cells together into what we call a pseudocell or a metacell. And then we would calculate the CNV score within that group of cells. Okay, so moving on to the next feature here, which is cell-cell communication. And this feature sounds exactly what the title is, where we are able to identify what cell types are likely to communicate with each other based off of the expression of the ligand gene or the receptor gene. So these are some of the visualizations that you can produce from BBrowserX Pro, where you can visualize these ligand and receptor interactions, as well as their associated pathways. Again, this tool is based off of the original CellChat package and utilizes the CellChat database to kind of infer the cell-cell communication. And lastly, here we have the pseudotime or the trajectory analysis feature. So this feature allows you to track gene expression changes across disease progression or across different cell states. So for example, one of the questions that you can ask is, okay, I have some naive and mature cell states within my data set, and I want to see what kind of genes are changing across the development from naive to a mature cell state. So pseudotime allows you to track that kind of gene expression. In addition, we also optimize the original package. So this uses the Monocle 3 package in Python to run the pseudotime analysis. But again, our team has made a lot of optimizations for it to run much more efficiently within the application. Okay, so let's jump into how you can actually run these tools on BBrowserX Pro and then how to interpret the results from there. So if you'd like to access this study that I'm going to be using in today's demo, you can go on to Talk to Data and search for this particular title here. This data set is going to be a colorectal cancer data set, and the authors were able to take samples from 3 different patients. Some that are some diagnosed with colorectal cancer, some that are pure cancers, and some that are healthy as well. Right, and a little bit about colorectal cancer here. So it's characterized by the loss of this APC gene. This loss of gene is found in 80 to 90% of colorectal cancers. And it's also characterized by the formation of polyps. So that's where the authors have taken the tissue from. It's from these particular polyps here. And what this image is showing here is a couple of pertinent genes that are important players in the development and the metastasis of colorectal cancer. All right, so let's jump into the first tool here, which is the cancer cell prediction. So you'll be able to find the cancer cell prediction tool over here on the left-hand side. And clicking this open, you can click New Analysis here. And there are 2 particular steps in which you have to define first. So the cancer type here is important to define so that the tool kind of has like a baseline for what type of cancer type your tissue is coming from. So for example, since this is colorectal cancer, I know that this is a carcinoma, meaning that the cells that are likely to be malignant and carcinoma are going to be the epithelial cells. And so in the second step here, the reference type, you can either choose to have the tool automatically choose the reference type for you, or to manually choose the reference type yourself. If I were to manually choose the reference type myself, you typically would have to choose a cell type. So since we know that in carcinoma, the epithelial cells are the ones that are likely to be malignant, that means cells like immune cells or stroma cells are the ones that are likely to be non-malignant in the tumor tissue. So for example, my B cells are a type of immune cell, and these are not likely to be malignant within carcinoma. Okay, and so once I've chosen my reference type here, I can click Run Analysis. And the results that we are going to get, I have a couple results here. So first, what you'll notice in the results page here is that the tool was able to kind of hard classify our data set into cancerous or normal cells. This is based off of a threshold that we defined here. And so this histogram that we're seeing on the right-hand side is the distribution of the predicted CNV scores. So we can conclude that cells with a high CNV score are likely to be cancerous. How do we actually determine what a high CNV score is? There's not really a definite threshold. So one way we make it easier for you guys to define that threshold of what a high CNV score is we use this particular method called Otsu's method to define the cutoff for you. You can also define the cutoff yourself by dragging this line right here, the separation, and that will change the classification of your cells over here on the left-hand side. So if I were to drag this threshold a little bit more to the right, then I will say that, cells with this CNV score or higher are likely to be cancerous. And you see that cluster of cancer cells kind of cluster over here on the left-hand side. All right, next, another result that we get is this heat map here. So this is where you're able to see kind of the spread of your predicted CNV scores, or the genes that are kind of upregulated in a pattern together or downregulated in a pattern together. You see in our cancerous population here, within, for example, chromosome 3, in this particular region, there's a lot of downregulation happening within chromosome 3 here. But when we compare that to our normal population, you see there's not really that much of a downregulation. Right, and this just gives you kind of an overview of, the spread of your CNV events happening. Okay, and then lastly, we have this phylogeny tab here. And this is where you'll be able to track how your different populations of cells kind of evolve into their cancerous state. So each of these little nodes represents a pseudocell or a metacell. And if we were to click on a node here, it'll be able to highlight those cells on the little UMAP on the right-hand side here. So for example, if we were able to kind of trace this lineage or evolution of cancer cells here, then I were to select out this population of cells, then I can see it kind of highlighted on the UMAP on the right here. Okay, and so yeah, that's how you're able to utilize this cancer cell prediction tool on BBrowserX, and also kind of interpret the results that you're seeing here. Additionally, the results will also be saved in your metadata too, so you can see the distribution of your CNV scores on your UMAP. Again, you can see that here, that the dark blue cells are the ones that are likely to be cancerous. And then some visualizations that I also want to show you guys here. This kind of is like a proof of concept of this using this tool. So when I looked at the distribution of CNV scores across the different sampling sites that the authors have taken the tissue from, I see that the cells with the highest CNV score appear primarily in the primary tumor. So this is to basically demonstrate that, okay, this tool was able to identify the actually cancerous cells that appear in our primary tumor, but not really anywhere else within our normal or a polyp tissue. Additionally, one of the ways in which you can utilize the CNV score is also by mapping the CNV score across your different subjects. And so you're also able to identify some patient-specific heterogeneity within cancer as well. So you can see here, this particular cancer patient, number 1, has a very high CNV score, or has a population of cells with a high CNV score, at least compared to patient 2 and patient 3. All right, and so the next tool that I want to introduce here is the cell-cell communication tool. So going back to BBrowserX Pro here, you can open the cell-cell communication tool on the left-hand side. And to run this, you'll first need to specify the cell type metadata. So I have these cell types here. And you can also split by different conditions if you have multiple conditions within your data set, which I do here. And what that will do is it will run the cell-cell communication iteratively across each of your different conditions. So I can choose to click run here. And let's view some results here. So here what you're looking at is a heat map, where on the y-axis you have your sender cell types, and on the x-axis you have your receiver cell types. And if I was interested in some particular interaction, for example, I'm interested in seeing how my colon goblet cells interact with cells that have a high CNV score, or in other words, cells that are likely to be cancerous. I can click on this particular node, and then it's going to open up another type of visualization where you can see exactly which ligand and receptor interaction is occurring between my goblet cells and my cancer cells here. And you can see that it occurs through this CLDN3 ligand receptor interaction. If I were to go back to my slides here, so yeah, this is basically what we're seeing. This occurs mainly through the expression of the CLDN3 ligand and CLDN3 receptor. And so basically CLDN3 plays a role in cell adhesion. So maybe these goblet cells are interacting with these cancer cells because they may be losing their cell adhesion, which is a really common characteristic of cancer cells. But I can even take this a step further and ask, okay, well, since I see this interaction happening and this cell-cell communication is based off of just my gene expression data, how can I actually verify my cell-cell communication results? And how can I actually know if these two cells, are communicating or interacting with each other? And this is where I can leverage spatial data to do this. So switching over here to our SpatialX dataset. So here I have a spatial dataset of colorectal cancer tissue. And for those of you who are unfamiliar, we also have recently released a cell-cell communication feature within SpatialX as well. So you can perform the same cell-cell communication within SpatialX here. So let me just open up the results here so we can see. So for example, if I were to examine the same cell pair, for example, I want to see how my colon goblet cells are interacting with my neoplastic cells or my cancer cells. I click on the node here and it gives me the list of ligand receptor interactions. And if I were to click on a specific interaction here, what you'll notice is that it's going to highlight the cells that are actually interacting. So this is really interesting to see. So the blue cells here are the neoplastic cells, and the red cells here are the goblet cells. And these lines that you're seeing are the actual interactions occurring between our different cell pairs. You can see here the goblet cells are sending a lot of signals to our neoplastic cells here, and they are quite proximal to each other as well. So this makes me hypothesize that, these goblet cells are the ones that are turning cancerous or developing into cancer. And so that's one of the ways in which you can leverage this type of, different types of multi-omic data to kind of add more to your results or add more to your analysis here. So again, just to recapitulate, I see this interaction happening within my goblet and my neoplastic cells in spatial data as well. And I see that the CEACAM pathway plays an important role in cell adhesion of epithelial cells. And yeah, I was able to identify that these neoplastic cells lose their adhesive abilities based on the CEACAM pathway. Okay, and lastly, to introduce this last feature here, pseudotime analysis. Let's go back to BBrowserX Pro here. So to run the pseudotime analysis, again, you'll click the pseudotime tool from the left-hand side, and you'll only need to define what t-SNE or UMAP you want to look at, and then click run. So I've done so here in this previous result. And what you want to do here for the first step is you want to select a route or define the starting point for your pseudotime analysis. So for example, I know that in my data set, I have 3 different conditions. I have a healthy condition, this FAP condition, which is the precancerous condition, and also the colorectal cancer condition. And you can see here how the colorectal cancer condition is kind of separated from our other 2 conditions. And I want to see the gene expression as the healthy and the precancers kind of develop into more of a cancerous condition. So I can select my root node from anywhere within this population here. So if I were to select this population here, what that is going to do is it's going to load our pseudotime graph here. So you'll see how as we begin from a pseudotime of 0, and as we progress through our cell states, then the pseudotime gets a little bit darker here. What's loading over here on the left-hand side is a list of genes that basically show how much the gene expression has changed from the start point to the end point here. And so this gene list does take a little bit of time to load. So I'm just going to summarize the results here in a slide. For example, if I were to select this mucin-2 gene from the left-hand side, you can very clearly see the pattern now of how mucin-2 is more expressed in our healthy and our precancerous population. But as we develop into a more cancerous population, then we start to lose that expression of mucin-2. And you can also see that represented down here in the pseudotime graph, where the mucin-2 expression kind of declines over the pseudotime. And so this what this is telling me is that, maybe there is a depletion of these epithelial cells, or maybe these goblet cells are actually turning into neoplastic or cancerous cells. And so that's why we are seeing this depletion of this mucin-2 gene. Okay, and so that's it for the Single Cell and Spatial Exploration Club this week. I will open up the poll here in a little bit. So I'd love to hear you guys' feedback on any of the tools that I presented here today. Let me launch this here. So yeah, a lot of these features were really advanced features, and I was able to cover 3 of them in 30 minutes. So I really want to see if you guys were able to kind of follow along and to understand what these advanced features do and how it can help you guys in your own analysis. I'm also opening up the floor to any questions that any of you guys have. So feel free to put questions in the Q&A as well. Okay, and then also in the poll, I'm interested in knowing which one of these upcoming features you guys are most interested in, because on our roadmap, we have a couple new features lined up, such as clonotype analysis or ATAC-seq analysis or support for FASTQ. So if any of you are performing this analysis and you'd like to see it on the platform, make sure to let me know, and this helps us prioritize which one would be on our roadmap first. Okay, we have one question from Shota who asks, are completely different algorithms used for cell-cell communication in BBrowserX Pro versus SpatialX? So this is a really great question. Because yes, we have cell-cell communication on both BBrowserX Pro and SpatialX. While they are both based on the cell chat package, one different thing about SpatialX is that it utilizes the spatial components in order to kind of calculate the interactions. So if I navigate over to SpatialX here, and I open up this cell-cell communication tool, two of the parameters that you can see that are specific to SpatialX are the radius and the minimum number of natives. So the way you can interpret this is the radius is kind of based on, the radius around a single cell and what you would count within that radius as a particular interaction occurring here. And similarly, for the number of neighbors, it defines it as the number of cells that are nearby that particular cell that can be counted within the calculation of the interaction here. So these two parameters here are the ones that are very specific to SpatialX and how it kind of leverages that spatial aspect of spatial data into the cell-cell communication calculation. Okay, so I hope that was a little bit more clear for you. So, okay, so I'll end the webinar here. Thank you so much, everyone, for joining this week. And feel free to reach out to our support email, support@bowtering.com if you have any other questions. Okay, thank you, everyone.